So the next part is traditional ancient Indian beverages. As I have already discussed, that basically it's a process of optimization. Whatever the steps they have followed, it's a process of optimization. And if we are talking about one of the beverage which is known as Kanji, traditionally Kanji was made through some vegetable extract or with the rice extract. इसीलिए इसमें जो टर्म आता है मड पाकम. So the main steps are for the preparation are boiling, decanting, cooling, then inoculation, starch breakdown, fermentation, gas formation, maturation. So these are the steps which have been discussed during ancient Indian period. But before we start, like we are starting with few of the grains. So if you're talking about the grains, we should understand what is the main constituent of these grains which we are dipping in water. So basically अगर हम राइस या मेज या इस टाइप का कोई ग्रेन ले रहे हैं तो उसमें जो मेन कॉन्स्टिट्यूएंट होता है दैट इज एमाइलोज एंड एमाइलोपेक्टिन्स. ऑफ कोर्स इट इज स्टार्च एंड वंस वी आर सेइंग it is amylose, so it is alpha 1,4 glycosidic bonds, and amylopectin is 1,4 and 1,6 glycosidic bonds at branched position. Of course their solubility is a little bit different. Normally the content of amylose is 20 to 30% and amylopectins normally 40 to 60% range. So whatever the initial changes is coming, they are coming in amylose and amylopectin. So the first step, that is the selection and soaking of grains process. So I'm saying we are taking the rice, millets, barley grains. हम normally चावल, बाजरा इनको dip कर रहे हैं and soaking it in water for 6 to 12 hours. So actually what we are doing, we are hydrating the starch, which is basically amylose and amylopectin. So if you are dipping it, it means after swelling what is going to happen. After embedded with water molecule, their hydrogen bond network is going to cleaved or damaged. Of course, there is not much chemical changes, but due to hydrogen bond network breakage, these compounds are going to be modified. Why? Because few of the enzymes which are found here, like amylases, alpha and beta amylases, proteases, they are going to produce, they are going to work, and the proper condition for them, because the hydration is taking place. So the cofactor metals, जो cofactor metals होते हैं और इनमें पाए जाते हैं, वो भी हाइड्रोलाइज होके फ्री हो रहे हैं. मतलब बेसिकली मिनरल्स और विटामिन भी लीच आउट हो रहे हैं, जो कि एक बेटर एनवायरनमेंट इन एंजाइम्स को नेटिवली काम करने के लिए देते हैं. साथ ही साथ ये जो ग्रेन्स होता है इनमें इनके सरफेस पे देयर इज द पॉसिबिलिटी दैट सम गुड माइक्रोब्स में आल्सो may increase, like lactic acid bacillus or some of the fungi, they may also grow. So if the proper condition will be provided to them, they may also grow. So in the starting process, basically we are breaking the hydrogen bond network, and as hydrogen continues, the function of alpha amylase and beta amylase is going to do good work. So once they started the processing, the next step is crushing or grinding. So basically you are doing mechanical disruptions. So mechanical crushing means you are disrupting the structural integrity of these grain tissue. So you are breaking the cell walls. So once the cell wall is breaked, basically the cell wall is made up of cellulose, hemicellulose and pectin. So you are giving the more surface area to interact with the water, and now starch is interacting with alpha amylase or beta amylase. The disaccharide and trisaccharides are going to convert like maltase and then further in glucose. Similarly, peptidic chains are going to change in amino acid, which are going to be a precursor for the required the microbial, because from them nitrogenous condition is of course the nitrogenous condition is required from them. The third step in which the boiling is taking place. So if we are doing the boiling, it means in this process the gelatization is taking place. In third step, the gelatization is taking place. So gelatization means the temperature is high, all the network has been breakout, the alpha amylase and beta amylase has converted all the starch molecule, approximately all the starch molecule, into the maltose and glucose. So once the temperature rise, it becomes a jelly like structure, little bit jelly, or we can say the viscous structure is going to prepare, and which intensified when it goes for the cooling. Now we are going from 60, 70 or 80 degree to 25 to 35 degree. So once we are reaching towards 35 or 40 degree, this environment is microbial friendly environment, and the pH is going to be acidic, which is more favorable for these microbes, or these suppose LAH, if we are talking about the LAH, or we are talking about some of the fungi which are required for the fermentation, we are providing the appropriate condition once the cooling phenomena is taking place. And of course, once it is cooling, the viscous nature becomes more intense in that case, and pH also reaches in acidic region. Now in the next step, once you have provided all the medium, natural fermentation is going to take place. So if I am saying natural fermentation, it is going to take place in two ways. The first is through lactic acid bacteria, which is LAB. So what lactic acid bacteria is going to do, it is converting one glucose molecule into two lactic acid molecules. So and similarly, if you are using yeast, it is going to change one glucose molecule into two ethanol molecules, or alcoholic molecule. So basically the fermentation is taking place. Similarly, protein is also breakdown, vitamin is leached out, and the other microbes which were also found, they will be inhibited, because the condition is more suitable only for these microbes. Once the fermentation takes place, you're going to add some of the chemicals or biochemicals in the form of vegetable and spices. If I'm saying vegetable and spices, one of the example is beetroot, and mustard seeds, which provides isothiocene cyanate salts, which gives a taste to this one. So basically, the purpose of addition of these vegetables or spices is one, the flavor, another one is taste. Taste comes out due to some specific salt, and flavor come out due to ester formation, because in the fermentation ethanol has been produced, and these vegetables or spices are going to provide the organic acid. So ester is going to be produced, normally the ethyl lactate or ethyl acetate, and due to these esters you're going to get some specific flavor, and after that this process continues and you get, the maturation. Finally the reaction matures and you get a stable organic acid profile, which is digestive and quite nutrient, and also probiotic in the nature. So basically they optimize the conditions in such a way that the carbohydrate, नॉर्मली हम आज की डेट में क्या कहते हैं कि कार्बोहाइड्रेट को अवॉइड करते हैं, करंट सिनारियो में आज की डेट में कहा जा रहा है कि कार्बोहाइड्रेट को थोड़ा सा अवॉइड करना चाहिए. दे वर वेल अवेयर. So what they were doing, they were taking carbohydrates in the form of starch, changing it in the form of gelatin. Then fermentation is taking place. After fermentation, it is more biocompatible or probiotic in the nature. With the help of some natural salts, vegetables, they are giving specific flavors, appropriate condition like acidic condition towards mild acidic condition, which gives it appropriate taste. So this was one of the method in which, if you are saying about the chemical reaction, so normally the chemical reactions are taking place. The first one in presence of, like, once you're putting this underwater for 6 to 8 hours under sunlight, so alpha amylase and beta amylase are breaking 1,4 and 1,6 glycosidic bond. So the enzymatic process. In the next step, starch is going to convert it into maltose and glucose molecules. Of course, protein is going to change in polypeptides and amino acids. Then the fermentation is another one. So during fermentation, two types of fermentation is taking place. One is LAB, in which glucose is going to convert it into lactic acid, and next glucose is converted into ethanol if yeast is used. So two alcoholic, the lactic acid and ethanol. So accordingly we are going to get two esters, one organic acid based, some vegetables or salt has been combined, few salts like like carotenoids, beetroots or mustard seed is going to be added in the before last step, so which will give some specific taste due to that salt characteristic. And after maturation, the conditions in mild acidic conditions, pH, it becomes more biocompatible, or you can say more probiotic in the nature, good flavored and good taste, and also more digestible and nutrient value. So this was the knowledge of our ancient India, which we should understand. So thank you very much.